In the previous video, we have understood the abstract of a paper where they are presenting a concept that under oxidative stress in the melanocytes, the calreticulum might migrate to the cell surface and then cause apoptosis of the melanocytes. So in order to validate those results, they did several experiments, and one of the figure is presented here. And the objective of this figure is to present that calreticulum overexpression is associated with oxidative stress. And the good thing about this experiment is that they did their experiment directly on the human patient skin samples. So first they check the calreticulum expression in the healthy epidermis and vitiligo patients, vitiligo PL epidermis, and they can find out that the calreticulum, this is the PCR expressions where they quantified this mRNA of the calreticulum, and they can find out that calreticulum expressed in this vitiligo is statistically significant compared to the case of this calreticulum mRNA expressed in this healthy epidermis. And after that, they need to check how this calreticulum increase is affected, how this calreticulum increase is occurred in this how this calreticulum increase is occurred in the vitiligo affected patients. So people might have different degrees of these skin disorders. So they check and this different degree of this vitiligo is been defined by this VASI score. So they check the relationship of this calreticulum expression with this different VASI score, and they can find out that this relations is directly related. That is, they are directly proportional to each other. So after validating that, with increase in these vitiligo disorders, there is an increase in the calreticulum expression. So they again did the immunofluorescence staining of this calreticulum and melanin A on the healthy skin and also this vitiligo PL skin. And this immunofluorescence staining results also shows that in the healthy skin the calreticulum expressions are lower compared to the case of this vitiligo PL skin, where calreticulum expression are higher. So calreticulum expressions are shown in the green fluorescence. So it is clear that green fluorescence are higher in the vitiligo PL skin compared to the sample of this healthy skin. And these things are quantified in the panel D, where we can see that this difference is statistically significant. That is, fluorescence intensity of calreticulum in the melanocytes in this healthy skin and this vitiligo PL skins are statistically significant, where vitiligo PL skins has higher calreticulum expressions compared to the healthy skin. After that, after confirming that in the vitiligo affected patients there is a high expression of this calreticulum, they need to check its relationship with oxidative stress. So in order to do that, first they check the oxidative stress expression in the wild type cells and also in the oxidative stress expression in the wild type cells and also in this vitiligo PL epidermis. No, not the cells, in the wild type healthy epidermis and this vitiligo PL epidermis. And they can see that H2O2 content is statistically significant in this vitiligo PL epidermis compared to the healthy epidermis. So once confirming that in the vitiligo PL epidermis both the oxidative stress are increasing and simultaneously the calreticulum mRNA expressions are also increasing, now they need to validate whether how this increase in H2O2 content is related to this calreticulum, whether there is a linear relations or it is related by some other mathematical equations. So when they plot this epidermal H2O2 content of the patients that have different H2O2 content and simultaneously calculate their calreticulum mRNA expression, they can find out that this increase in H2O2 content seems to be directly related to this calreticulum mRNA expression, where these patients that have more oxidative stress will have more calreticulum expressions. So from this whole experimental figures, they make a point that increase in calreticulin overexpression is related to the oxidative stress in the melanocytes or the patients who are affected with this vitiligo disorder, skin disorder. So we will continue our discussion about this calreticulins migrate to the cell surface and then trigger apoptosis of the melanocytes through immune response in our subsequent videos by discussing the other figures that are presented in this paper. Meanwhile, if you need to understand this paper in details, like how the experiments are conducted, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.